Okay, yeah, so good afternoon, everybody. My name's James. I'm a second year PhD student at Warwick in the School of Life Sciences. My PhD is predominantly microbiology, specifically looking at drivers of antibiotic resistance in the environment, particularly in river systems based in the UK. So I appreciate I've got quite a mixed audience today. So just to sort of put things through before we carry on, when I talk about antibiotics, I refer to sort of any chemical substances that can kill or inhibit the growth of bacteria. I'm sure many of you have had to take antibiotics either given to you by a GP or used in a hospital, for example, to treat various infections. And when I refer to antibiotic resistance, I then refer to, in broad terms, the ability of bacteria to resist the effects of an antibiotic to which they will want sensitive. And there are a number of mechanisms by which bacteria do this, but most of generally revolve around bacteria acquiring antibiotic resistance genes. And this occurs as a result of the exposure to of an antibiotic, a bacteria to an antibiotic over a period of time as a selection pressure. So why antibiotic resistance is such a major issue? I think this sort of figure really puts things forward here, and it's quite useful in explaining that. This data was commissioned by the UK government in 2014, and it estimates that by 2050, if we're not doing a sufficient amount to counteract antibiotic resistance, we could see up to 10 million deaths a year worldwide attributed to it. And that could largely be a result of people having routine operations not working and people picking up infections, which is really worrying. And when you think obviously about the current sort of COVID-19 pandemic, unfortunately it's killed nearly sort of 900,000 people worldwide now. You can imagine the enormous impact this would have. So it's really important that we've got good research going into it. And I'm sure many of you will have seen campaigns such as these commissioned by the Department for Health, trying to decrease the amount of antibiotics that are prescribed, but also the amount that are taken inappropriately by individuals. And I think one element as well that's not quite so well understood is the amount of antibiotics we actually use for animal purposes. So you can see there just over 25% used for farm animals, just under 3% used for our pets. So it's important to understand all of these dimensions of where antibiotics are used so you can understand where and how antibiotic resistance is arising. So I mentioned at the start of the talk that sort of my PhD project and my research group look at antibiotic resistance in the environment. And I think, again, this figure is quite useful in explaining how antibiotic resistance occurs there and why. So as you can see here that both humans and animals will consume antibiotics for various purposes to treat infections. And there is the possibility for the transmission of both the antibiotics as a chemical, as a drug, and also antibiotic resistance genes that are carried by bacteria to be transmitted between humans and animals. And a big example of that is if you consumed meat, for example, that came from an animal that had been exposed to antibiotics over a long period of time, the likelihood is that you will ingest that. And in many cases, our digestive system is not able to break down the antibiotic, so it will remain in our system and it will be dispensed in our waste. And of course, in the UK in particular, the majority of our waste is treated at large wastewater treatment plants, which will in theory clean the waste and try to get rid of most of the bacteria that are present there. But there is a growing body of evidence that suggests that the processes that we're undertaking at the moment aren't sufficient. And so you have both antibiotics as a drug and resistance genes getting into river systems when the effluent of the treatment plant is released, which is of real concern. But in the same way, of course, the antibiotics that we use on farms, for example, there is potential for the drug to be maintained in animal manure. And as a result, during periods of high rainfall, for example, you can see the wash off of this manure into the soil system and also into river systems as well, which is of concern. But mainly in our group, we look at the impact that sewage treatment plants have on the accumulation of resistance in the environment. So in the UK, these will process millions of litres of tons of human waste every day, and they will release their treated effluent into the environment directly. And why this is of such a sort of big issue is that when you actually look back at the antibiotics that we've used and mass produced as humans, the first generation of antibiotics that we produced, over 60% of them that were used for clinical purposes were derived from a particular bacterial species known as Streptomyces, which was initially isolated and found in soil. So antibiotics are natural products that are produced by bacteria in the environment as a defense mechanism for themselves. And we've exploited those properties for medicine over a period of time. So understanding the environmental dimension of resistance is really important because it can directly impact people's health. And I think recently we've seen a number of high-profile cases of water treatment companies, particularly in England, being fined large amounts of money by the Environment Agency for what were deemed serious failures of their policy and their processes. So you can see on the left here, Thames Water will fine 20 million for a large raw sewage spill, and you can see the sewage just like accumulating on the surface of the water. It's pretty gross. Again, Southern Water being fined a hefty amount for another serious spill. And these companies receive a great deal of government and public funding. So it's really vital that's sort of held to account, and we sort of know that their processes are kept up to date and working. So in terms of my PhD, there's sort of three broad aims that I'd like to sort of very slightly put across to you today that we're looking at. So the first one is I was carrying out a targeted analysis on some very large data sets that were derived from a previous project in my lab group, which focus on river sites across the River Thames region, which has revealed some interesting results that I'll talk about later. I'll talk about briefly some of the experimental work that I do to look at the impact of the antibiotic residue that's found at river sites and the impact it has on the accumulation of resistance. And then the last bit, I aim to look at to determine the genetic context of antibiotic resistance in the environment. And we try to use this to determine whether or not there are individual bacterial species that are particularly associated with different types of resistance and how we can determine that. So as I said, the sort of first aim of my sort of PhD project was to carry out sort of targeted analysis on these large range of sites on the Thames catchment region. And what you can see here, there's 69 sites in total, and the sites really do range in terms of their location, their geography, their land coverage, and the population that's associated with them. So what happened with this is that sediment and water was collected from these sites. DNA was purified so bacterial DNA was purified, and a technique known as quantitative polymerase chain reaction basically was used just to determine the different levels of antibiotic resistance genes that were present. And here's just a list of a few of the ones that were investigated here. And I wanted to combine this with a range of ArcGIS data relating to the land use of sites, and also chemical data which showed the antibiotic concentrations that were present there. I also had a great deal of what we call metagenomic data, and for those of you who aren't in microbiology and won't have a clue what that means, in basic terms, it just means we collect samples from the environment, we extract bacterial DNA from them, we then sequence that DNA, so we just get a big list of the genetic information that's contained there, and we carry out sort of annotations to look for individual sequences of a gene that we're interested in, so one that's associated with resistance. And from this data set, we had just over 1,000 potential antibiotic resistance genes that come from the environment to look at, which is quite a hefty amount. And to try and sum up all of that sort of analysis in a 15-minute talk just isn't going to happen. But I think this correllogram is quite useful in just demonstrating some of the broad trends that we've come across as part of the analysis. So in this correllogram here, you've got labeled in blue individual drugs. In red, you have antibiotic resistance genes. In the yellow, you've got metagenomic data, and in the green, you've got the land use of individual sites. And from the red sort of dots you can see here, these are positive correlations. So, for example, you're seeing positive correlation between the presence of certain drugs in the environment and antibiotic resistance genes, which you might expect. There's also quite interestingly a correlation between those sites which are most urban, suburban, which I show here, and the presence of drugs in the environment. But also, interestingly, from this one negative correlation you see here, this is 60 So this is the overall bacterial communities, that the makeup of different bacteria that are present in that sample. So interestingly, in the presence of the various drugs, you generally tend to have a negative impact on the bacterial community, which means that the drugs are potentially having an impact and potentially killing off some environmental species. In terms of just some of the experimental work that I do, I carry out what we call microcosm experiments in microbiology. So basically, I go to a site that's potentially downstream of a wastewater treatment plant, a river site, and we go and collect sediment and water, and basically just bring it back to the lab. So this is me setting up an experiment here. And we bring them back, and we just basically set up like these systems, like small aquariums in the lab, whereby we try to mimic the conditions you might have in a river. So we'll have a layer of sediment and a layer of water. And over a particular period, so we've done experiments over 25 days, we'll measure the physiological conditions, we'll measure the temperature, we'll measure the pH, the oxygen, to have a look at how that changes. And what we'll do is, for 3 of these tanks out of the 6 that you see in person here, I'll input an antibiotic at a certain concentration. And that'll be a concentration that's far below what you'll ever find prescribed in a hospital or by a GP, because these are sort of consistent with some of the concentrations that we see in the environment. And we aim to look at the direct impact that has on the accumulation of resistance. So we'll take samples of water and sediment every couple of days and analyze them. And I carried out one of these experiments on a site which was involved in quite a high-profile case of sewage spillage by, again, Thames Water, where they were fined just over 600,000 a few years ago at a site that's just south of Maidenhead and north of Bracknell. So it was pretty horrendous. They released millions of litres of raw sewage into the stream, which resulted in the death of hundreds of fish. And this is some pictures of the site and what it looks like today. So we went back to sample there, and you can see the sort of formation of like sort of sewage, like gunk on the surface of the water. The colour's pretty bad, it smells pretty bad. It just shows you the sort of conditions of some of our rivers that we have in the UK and how bad they can be. And from the experiment that we carried out on that particular site, this is just an example of some of the data that we were able to generate. So what you have here is an individual antibiotic resistance gene. So this is SOL1. And you can see here that in the presence of sulfamethoxazole, which is an antibiotic, in the presence of the drug in the system, you see an exponential increase in the prevalence of the antibiotic resistance gene, which is not reflected in the absence of the antibiotic. So in addition to those correlative sort of impacts that we saw in the previous slide, this gives evidence of a direct causative impact of the presence of the drug, which shows you that those concentrations you might find downstream of a treatment plant are directly impacting on resistance. And just to sort of back that up, there's a little bit more data just to demonstrate that, again, in the presence of the drug that we see here in green, the dissolved oxygen concentration over a 25-day period was notably higher than it was in the absence of the drug. And this is probably largely due to the drug having an impact on the bacterial community, even at those low concentrations, potentially killing off some environmental bacteria. And as a result, the demand for dissolved oxygen in the presence of the drug will be lowered because there's less demand for bacteria to undergo respirative processes. So that just further demonstrates the impact that the drug has in our system. So the sort of broad conclusions that we've been working on and carrying on moving forward is that the presence of a sublethal antibiotic concentration can have a significant impact on the prevalence of antibiotic resistance genes, even if those concentrations are very low. The sublethal concentrations can have an impact over a pretty short period as well, and that the antibiotic can also have a big impact on the physiological conditions, which will affect the bacterial community, and potentially even other wildlife that are present there. And just very quickly, just finish off. So I've seen the sort of correlative impacts of sort of between the presence of drugs in the environment and the accumulation of resistance, as well as the causative impact. But what we don't quite understand is whether or not there are particular bacterial species in that environment that are accumulating resistance more readily than others. And because there's so many bacteria out there, and if I refer back to Vincent's talk earlier on today, the community of bacteria that you have in a gram of soil is enormous. So to try and appreciate all that diversity is really difficult. So we basically want to use this method known as Hi-C, which has been used previously to look at the assembly of bacterial genomes. And what we want to do with it is it allows you to look at the 3D distances between individual gene sequences inside cells, and could potentially give you direct evidence of DNA sequences originating from the same cell. And what we want to do with this is to combine this Hi-C analysis with our previous data to associate antibiotic resistance with genomes, whether they be plasmid or chromosomal, and try to understand the transmission routes of antibiotic resistance into the riverine microbiome, and establish essentially how antibiotic resistance genes are being disseminated from a bacteria that might carry it into natural indigenous bacteria. So that's it from me today. I'm happy to take any questions. Thank you very much.